Hello students, welcome to my channel. The topic for discussion is mapping of genes by interrupted mating technique. Before going to the topic, please subscribe my channel. Press the bell icon for notifications. Like, share among your friends and comment. You should know what is high frequency recombination or HFR strain in bacteria. When F plasmid or sex factor integrated with chromosomal DNA, then such bacteria is known as high frequency recombination or HFR strain bacteria. In the cross or conjugation between HFR cell and F minus cell, frequency of recombination is very high, but frequency of transfer of whole F factor is very low. The diagram shows formation of HFR strain when the F plasmid is integrated into the bacterial chromosome. See here in this diagram, chromosomal DNA and F plasmid is shown that that is called as donor. In conjugation we already studied. Donor or F plus strain. Then integration of F plasmid into bacterial chromosome DNA take place and HFR strain or high frequency recombination cell is formed. In the integrated state 2, the genes of F factor are functional and can bring about the formation of the F pilus, replication and transfer of the plasmid in the integrated form. However, the transfer of the entire integrated F factor requires a long time and hence happens at a very low frequency, that is 1 in 10,000 due to random breakage. Therefore, in most of the cases, only the initial part of the F factor, that is adjoining origin T, adjoining ORIT and few bacterial genes and not the entire integrated F factor are transferred from the HFR strain into the F cells. The donor chromosomal genes transferred get incorporated into the genome of the F minus cell, resulting into the recombinant cell. Hence, the recipient a F minus cell is mostly not converted to F plus cell but a F minus recombinant cells. See, all these are given in these 5 diagrams. 1, 2, 3, 4, 5 is given. Conjugative transfer between HFR and F minus cell is shown. Next is linkage determination using conjugation. More information on conjugation were obtained by experiments conducted by Eli Wallman and Francois Jacob. In one of their experiments, they mixed 2 strains, the HFR, that is H, isolated by Hayes, and a F minus recipient cell in liquid media favorable for growth of both the bacteria. The bacteria were of the following genotypes. HFR, that is STRS, AZIR, TONR, LAC plus, GAL plus. F minus, another strain, STRR, AZIS, TONS, LAC, LAC minus and GAL plus. Here the abbreviation is given. STRS is sensitive to streptomycin, AZIR resistant to azithromycin, resistant to LAC plus, could utilize lactose as sole source, GAL plus could utilize galactose when solo source of carbon. The conditions favorable for the conjugation were provided. After a few minutes, the bacterial cells in the liquid media were agitated in a kitchen bladder to separate the paired cells, or in other words, to interrupt the conjugative mating. This procedure is called interrupted mating. These steps were repeated at regular intervals from the initiation of conjugation. The cells were then transferred onto the selective medium containing streptomycin to remove the streptomycin sensitive HFR donor cells. The cells growing on the media containing the streptomycin resistant cells were then investigated for other genes. The streptomycin resistant recipient cell have a gene from the donor or the one in which conjugation had initiated. These cells are called X-conjugants. In this experiment, they observed that the recombinants gain AZIR gene after around 8 minutes, TONR after 10 minutes, LAC plus after 18 minutes and GAL plus after 25 minutes. Also, the percentages of ex-conjugate colonies were 90% for azithromycin resistant, 80% for TONR, 40% for LAC plus and 20% for GOL plus. See, that is the figure is given here. See in the figure. Hence, each gene entered the F minus cell at a particular time, and the percentage of the cell receiving the early entering genes were much higher than the cells with the later entering genes. These observations lead to the conclusions that the transfer of the HFR initiated at a particular point. Hence, each gene was transferred at a particular time after the initiation of the conjugation. This point of initiation is termed as origin of transfer, that is ORIT. The transfer of the genes took place in a linear manner. Hence, the later genes were transferred to a smaller number of bacterial cells compared to the initial genes at the mating pairs were separated during the transfer process. These experiments made it clear that such experiments could be used to predict the order of the genes in the bacterial chromosome. Hence, a bacterial chromosome map can be prepared and the distance between the genes can be measured in terms of the time in minutes of the chromosome transfer. Wallman and Jacob constructed linkage map from the interrupted mating results at the time without knowledge of the physical basis. Example, in the figure, bacterial map with the order of the genes entering the F- recipient cell with respect to time are mapped using data from figure 4 that is given below. Chromosome circularity and integration of F factor. Interrupted mating technique also gave early evidences of the bacterial genome being circular was obtained, which has been confirmed by various other studies later. It was observed that when interrupted mating studies were performed on different HFR strains, the sequence of the genes entering the F- cells varied considerably from strain to strain. The time at which a particular gene entered was not fixed, however, it always entered between the same adjoining genes. For example, in the figure, the sequence of genes vary, but the gene PUR is always between LAC and GAL. Alan Campbell concluded that this was because the chromosome of F plus donor was circular. As the HFR chromosome is generated, when the circular F plasmid gets integrated into the circular bacterial chromosome, the difference in the sequences of the genes in the different HFR strains arise due to the difference in the location and the orientation of the F factor insertion. As the transfer always begins from the origin of transfer and the location of the F plasmid insertion determines where transfer begins. Hence, the adjacent genes vary and so does the sequence of gene transfer. The orientation of the plasmid within the bacterial chromosome determines whether transfer will occur clockwise or counterclockwise. Hence, the conjugative transfer in HFR cell can be subjected to interrupted mating technique and used to map the entire circular bacterial chromosome. See in the figure, the location of insertion and orientation of the factor determines sequence of the genes transferred. See, everything is marked. Fertility factor, origin, that first to enter, then terminus, last to enter. Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.